Hello, welcome back from your break. So now that we know how the somites are formed, I would like to talk to you about how the presomatic mesoderm is established, and in doing so, also talking a bit about the axis elongation. So how the axis, or if on the embryo, become a bit longer. So this is mostly due to the tail bud elongation, and most of the information, and here is a sketch or a representation of a zebrafish tail bud. In fact, zebrafish have been fundamental for our understanding of how the tail bud, or if you want, the axis is elongated. And it is a region, the tail bud, full of cells that are doing different things. So it is a very important region for axis elongation and for cell fate determination. Indeed, we have a region that is quite dorsal in the tail bud that is called neuromesoderm progenitor region, or NMP. This region here is called neuromesoderm progenitor region because the cells in this region that are moving, as you can see from the arrow here, they're moving posteriorly. They can either become neurons, so become part of the neural plate, or become part of the presomatic mesoderm, so become mesoderm. So they're called neuromesodermal progenitor cells. In the tail bud, we recognize 4 regions where cells are doing different things. The first one is the DMZ, the dorsomedial zone. So DMZ is not for demilitarized zone, but is the dorsomedial zone. The cells in the dorsomedial zone are moving posteriorly. Okay, so are pushing the posterior part, are moving posteriorly, and doing so pushing the posterior part of the tail bud. Then the cell from the DMZ enter what is called the progenitor zone. The cell in the progenitor zone move laterally, both right and left, and also move centrally. So the cell from the DMZ that move posteriorly, then decide whether to move laterally, left or right, or move back deep inside the embryo, but centrally, okay, following the arrows. Now, the cells that move laterally in the maturation zone will become mesoderm. The cells that move internally, if you want, are going to form in the maturation zone as progenitor for the neural plate. And then we have the last part is called the emergent zone, or where the presomatic mesoderm is defined if the cells are moving laterally, and is where the cells that move centrally are going to be part of the neural plate and therefore the neural tube. So during this process of moving of the cells, moving laterally and posteriorly, and then internally, we have elongation. We have basically elongation of the tail bud. So it is this collective migration of cells that push the tail bud posteriorly and elongate the embryo itself. It is important at this point to notice that the cells that are moving laterally express different genes than the cells that are moving centrally. This is, it should not be a surprise for you, because the cells that move laterally are going to become mesoderms, while the cells that move centrally, they're going to become neural plate. So the cells that are moving laterally are going to express genes like MSG1 and TBX6, while the cells that are moving centrally are going to express genes like SOX2. Now remember, we already mentioned SOX2 when we were talking about neurulation. So this is not something new to you. You know that neural plate cells, or cells that are important, that are dedicated to the formation of the neural tube, express genes like SOX2. Moreover, here, during the maturation, in the maturation zone, cells undergo a cell division. at least one cycle of cell division that is controlled by genes like CTC. So gene expression of these genes controls cell division in the maturation zone. Okay, now we talked just now about how the axis is elongated in the tail bud, and how the tail bud itself is elongated by the migration of these cells. But how do we stop this elongation? At one point, the tail bud needs to stop growing, okay? Otherwise, the embryo will become infinitely long. So how do we stop this growing? It is very interesting and very elegant. Indeed, the OX genes are important for the stop or inhibition of the tail bud growing. These are examples that I've been studying a lot in xenopus. So if you remember just a few slides ago, we talked about this collinear activation of the OX genes. We say that the more posterior OX genes are expressed later during the development and in the more posterior part of the embryo along the anterior posterior axis, right? So we also were saying that the cells that enter the primitive streak late during the development, if you want, and more posteriorly are going to express more posterior OX genes. Well, it turns out that many of the most posterior OX genes have a repressing activity of wing signaling. So the more posterior the OX genes are, the stronger repression they have for wing signaling in the tail bud. So so at one point, the cells that are entering the DMZ are starting to express a lot of these very posterior OX genes that have a very high level of repression from wing signaling. And if you remember, wing signaling is connected to the FGF. So the two of them control, if you want, reinforce each other's expression. So when we start to remove wing signaling because we have these OX genes that are now repressing that signal, the tail bud growth starts to slow down and it is inhibited. Okay, so this OX signaling start the inhibition process of the tail bud growth. So what's happened now? When the tail bud start to be inhibited in its growing, there is less cells that become presomatic mesoderm. And in a way, the somite formation become much quicker than the tail bud growth because we are inhibiting the tail bud growth via the OX genes, right? So in a way now, the somite formation start to exhaust all the existing PSM, presomatic mesoderm. And what's happened from the point of view of expression? If you remember, we said that the neural plate cells, the neural tube cells, but also the somite cells express retinoic acid. Okay, and we said that the retinoic acid inhibit expression and activity of FGF. All right, so we have now that somites are growing more and more. We have more and more neural tube that express more and more retinoic acid, and this retinoic acid is repressing FGF. On the other hand, the OX genes of the cells that are entering the primitive streak now are repressing the wing signaling. So now this feedback loop between wing signaling and FGF is really weakened a lot. At one point, if you remember, we said that FGF induced repression of retinoic acid by this kind of inducing the expression of these repressors like CYP26, right? But then at one point, the level of FGF is so low that it cannot anymore induce the expression of this CYP26 repressor. Therefore, when we remove this repressor, we have an hyperactivation of retinoic acid. So when we have this hyperactivation of retinoic acid, that's it. The FGF wing signaling is shut down completely, and there is no more tail bud growth. So at the end of the day, the retinoic acid gradient wins against the FGF gradient. Fine. Now, we talked about the somite formation, so from the presomatic mesoderm. But we now need to talk a bit more about how the different parts of the somite form and differentiate in the dermomyotome and sclerotome. So you see here a representation of somite near the notochord and the neural tube, and part of the somite are going to form the sclerotome, while part of the somite are going to form the dermomyotome. Now, how the cells in the somite are able to know whether to become sclerotome or dermomyotome? Well, it is because of signaling from, for example, the notochord and the neural tube. In particular, signals like SHH and NOGI. We know that SHH and NOGI are expressed in the notochord and the neural tube. Okay, we already talked about that. On the other hand, the ventral part of the somite, okay, the ventral part of the somite receives signals from the notochord and the neural tube, SHH and NOGI, while the dorsal part of the somite receives wing signaling, a BMP4 signaling, mostly from the neural tube and the epithelium. If you remember, when we were talking about neurulation, we say that when the neural tube is going to form the different neurons, in the neural tube there is a gradient of expression of BMP signals from the dorsal part of the neural tube and SHH signaling from the ventral part of the neural tube. So if you don't remember, go and have a look on that lesson. So the dorsal part of the somite receive from the neural tube mostly BMP and wing signaling, and it receives this signaling also from the epithelium, while the ventral part of the somite receive mostly SHH and NOGI signaling from the ventral part of the neural tube and notochord. So the ventral part of the somite, because of this SHH signaling, mostly become sclerotome. The dorsal part, on the other hand, is going to form the dermomyotome. Noggin is also important because it inhibits BMP4 signaling from the lateral plate mesoderm, and this is extremely important for the formation of the cartilage. In particular, SHH is important for the formation of the cartilage. Remember, the cartilage is actually part of the bone formation, and remember that the sclerotome forms basically bones. Okay, so we mentioned how the cell in these somites are able to differentiate the dermomyotome and are able to differentiate the sclerotome. So what are the signals that are important for these two? But you remember we said that in between the sclerotome and the dermomyotome there is the syndrome, and the syndrome need to form the tendons, right? So what are the signals that are important here to make sure that these cells knows that need to become syndrome? Well, the cell that defines syndrome is scleraxis, okay? So scleraxis is the gene that is expressed in the cells that need to become syndrome, okay? Scleraxis is actually controlled by a number of transcription factors that are activated by FGF, and this FGF is produced by myotome. So the myotome, which is part of the dermomyotome, express FGF, and these FGF signals induce a number of transcription factors which activate scleraxis in the cell that need to become syndrome. Okay. Here it is important to notice as well that the cell of the sclerotome, on the other hand, express a number of SOX genes, okay, SOX protein if you want, and this SOX protein in the sclerotome inhibit the transcription factor that otherwise will induce the production of scleraxis. So in this way, the cells of the sclerotome avoid to become syndrome. And the differentiation between becoming syndrome and sclerotome, SHH play an important role. Indeed, SHH induce the expression of the SOX group of genes, that inhibit the expression of the transcription factor that would induce the expression of scleraxis. So in a way, SHH maintain the identity of the sclerotom cells, avoiding the expression of scleraxis that otherwise will turn the sclerotom cells into syndetome. Okay, we talk about today so far about the paraxial mesoderm. We talk about the formation of the somite, and now the somite, the different parts of the somite are going to produce muscle and bones. Okay, so because we talk about the formation of muscle and bones, I think it's important now to take a few minutes to discuss how bones and indeed muscle are formed. So I'd like to talk to you about endochondral ossification. So the endochondral ossification is responsible for the formation of most of the bones of your body, apart from the skulls and some other bones. But endochondral ossification is the transformation of mesenchymal tissue in cartilage, and then this cartilage will become bone. So initially, this mesenchymal tissue will form a cartilaginous template, and these cartilaginous templates then will become bone. This endochondral ossification is typical of the vertebrates, the ribs, but also the bones in the limbs. Endochondral classification has 5 stages. The first one is called commitment. So during commitment, the mesenchymal cell commit to become cartilage, okay? And this is driven by SHH. We just talked a minute ago about how the SHH is important to differentiate between the syndetome, so the cell that become the tendons, and the sclerotom, the cell that will become bones, and initially they will become actually cartilage. After the initial commitment, you have compaction. So the next stage is compaction. The cells that are committed to become cartilage and therefore bones will compact, form a, if you want, a condensed compact modules. Okay, the cell of this compact module have different roles. So the most internal cells will generate the cartilage, while the most external cells tend to become bone. Here, this should not surprise you, but some of the transcription factors that are important for the formation of the cartilage are SOX, like the SOX genes, and in particular SOX9. SOX9 is one of the proteins that is extremely important for the formation of the cartilage. It induces the expression of collagenol and aggrecan that are extremely important for the formation of the cartilage and the cartilage function. So again, you see that we mentioned SHH a few times. SHH is one of the fundamental proteins, fundamental signals that induce and maintain the identity of the sclerotom, and is one of the important signals that induce the expression of SOX9 that is fundamental for driving the expression of genes that define cartilage functions. After this, the following step that we have is proliferation. So the chondrocyte are the cells that are now committed to become bones and cartilage. So this cell, the chondrocyte, proliferate. So they divide rapidly and they form what is called a cartilaginous model of the bone. So here is a sketch, an example of this proliferation. And while there is this proliferation, you have a formation of the bone template, if you want. But this, at the moment, is made of cartilage. In fact, this chondrocyte, during proliferation, also produce extracellular matrix that is cartilage specific. So it is a cartilage specific extracellular matrix. Soon after that, we have what is called an hypertrophic step. So the chondrocyte, especially in the middle, if it's a long bone, but not all bones are long like that. But anyway, if you're talking about a long bone, the chondrocytes, especially in the middle, stop growing, and they become what is called hypertrophic chondrocyte. What does it mean? Well, they stop growing and they become bigger. So they change volume, they become much bigger cells, right? When they become much bigger cells, they start to become bones as well. So they become the process of ossification. One of the first things that they do is to produce, they change the extracellular matrix. They begin ossification or calcification, most importantly, actually most precisely, calcification of the external matrix by deposition of calcium phosphate. The last step after that, we have the formation of the hypertrophic chondrocyte, is that these chondrocytes start to die by apoptosis, and the bone cells start to form in substitution of the chondrocyte. So what we have is that the hypertrophic chondrocytes start to die by apoptosis. So you can see here in the middle, these yellow cells here are the chondrocytes that start to die, and they have been substituted by osteoblasts. Osteoblasts are those cells that are going to form the final bones. The hypertrophic chondrocytes are replaced by these osteoblasts, which are the bone cells, and when this is happening, the hypertrophic chondrocyte, while they are dying, they send signals that induce the formation of blood vessels. So we have an invasion of the forming bones, if you want, of new blood vessels. So there is a proliferation of this blood vessel inside the bones. Now the osteoblast now begin also to change the matrix, and the cartilage, they become the ossification of the matrix that was mostly cartilaginous before. So this cartilaginous matrix that actually was it was added, it was start the process of calcification, now become bones when the new osteoblasts are invading the template. With the blood vessel, new type of cells come in. They're called osteoblast and osteoclast. These osteoblast and osteoclast are important to digest the apoptotic chondrocyte, and they form a hollow, if you want, hollow in the middle of the bone where the bone marrow will grow. So they form the bone marrow cavity. So in the final stage of development of the bone, you will find a region at both sides of long bones where we have proliferating chondrocyte, then hypertrophic chondrocyte that are going through the process of dying and being substituted by osteoblast, and the osteoblasts are going to form the final bones. Okay, so we have almost done. The last thing that I want to mention in this lesson is, the last things that I want to mention in this lesson are the formation of muscle. So the cells that are going to form muscle, so if you want the muscle cell precursor, are called myoblasts. Okay, the myoblasts need to form multicellular, multinucleated muscle fibers. So the myoblasts are going to produce muscle fibers, and the muscle fibers are structures that are multicellular and multinucleated. So the myoblasts need to produce these structures. How do they do that? Initially, the myoblasts, you can see them here, align themselves in long chains, and they express membrane glycoproteins so they can actually bind to each other. Okay, so form long chain that bind to each other. After that, the myoblasts express meltrin. Meltrin is a protein that induces membrane fusion. Okay, and so therefore the cells in the long chain fuse together and form long tubes or myotubes that are multinucleated. Meltrin also induced the expression of myogenin. Myogenin is important because myogenin controls the expression of muscle-specific genes. So now when myogenin is activated, the cells become completely, the cellular defect is completely sealed and they only produce muscle cells, muscle fibers. So after this initial fusion, the fiber, if you want, the initial myotube, need to grow and form strong fibers. So they go through a process of even more fusion. So there are fusion with neighboring, if you want, tubes or isolated cells. And then this process of fusing with neighboring cells or other myotube increase until they form strong final fibers, muscle fibers. The process of the formation of the myofibers via the fusion is controlled by interleukin-4. So interleukin-4 control this fusion process after the after the initial expression of meltrin. So after the initial expression of meltrin, interleukin-4 induce more fusion and growth of the muscle fibers. So the EL4, interleukin-4, recruit more myoblasts, more myotube, and allow the growth of the myotube in a muscle fiber. Contrary to interleukin-4, myostatin control this growth negatively, so reduce the fusion and reduce the growth of the muscle fibers. Indeed, there is a balance between the expression of EL4 and myostatin to control how big the muscle need to grow during the development. We can find in agriculture, for instance, myostatin loss of function mutants. So some animals do not express myostatin or express very low level of myostatin, and therefore their muscle fibers grow exponentially. They have what is called hyperplasia and hypertrophy of the muscle. They become what is called Herculean animals. This is a nice example of Herculean animals. These two animals are the same species. These two dogs are the same species, the same breed. This one is a wild type. This one is a myostatin null function mutant. Now, null function mutant are naturally occurring, so you don't need to do anything to the animals to present it. In agriculture, this kind of animal have been bred a lot, not only for dogs but also for bull. Some bull are myostatin loss of function mutant, and they are absolutely gigantic. They have a lot of muscle fibers. They look almost like they're exploding. Okay, so because I'm a bit weird and I keep thinking about this stuff even when I watch movies, the first time that I saw Captain American movie, when they transformed Steve Rogers in Captain American did, I thought whether some component of the serum that they used to transform Steve Rogers in Captain America was like a mutant agent that would change the expression of EL4 and myostatin. So if you reduce myostatin with a null function, loss of function, and overexpress EL4, you end up having a person like Captain America that is full of muscles. Okay, so at the end of this lesson, you should be able to discuss and explain how the paraxial mesoderm is produced and produce somites, and how the somites produce their derivatives. Okay, well done. We have finished for today. Take a break, perhaps revise your notes, and please take quiz number 2 of this lesson.